लास्ट टाइम से मुझे लग रहा है प्रकाश से ज्यादा कैंडिडेट है, ऐसा मुझे लग रहा है ना।
Yeah, I'm also seeing this time little more.
हम्म।
Maybe I think topic is also
करेक्ट। या।
interesting today for them. माइक्रोबायोलॉजी में ड्रग डिस्कवरी है तो क्या है जानना चाहते होंगे।
हाँ।
Some 1 month back, actually, on similar line, there was one webinar
हाँ।
organized by Satyabama Institute of Science and Technology on drug discovery.
So, good evening, everyone. On behalf of the Roundtable Discussion Committee of the Microbiology Society, India, I warmly welcome all the participants, students, researchers, faculty members, and professionals joining us today for the enriching session on the drug designing, discovery, and development. So, let me begin with the short introduction for this topic. So, now we know that the journey from the identifying a disease target to developing a safe and effective drug is one of the most fascinating, we can say, or the challenging areas of the modern science. So now the drug discovery is not limited to the traditional laboratory, means the wet lab experiments only. Today it integrates the biology, chemistry, bioinformatics, even artificial intelligence, machine learnings, and also the clinical, it's also accelerates the development of the novel therapeutic drugs. So today's discussion aims to provide the insights or the knowledge sharing into the various stage of the drug discovery, including the target identification, lead generation. We can discuss about the molecular docking, optimization, preclinical strategies, clinical trials, and also the regulatory approvals or the policy. So we hope this session will inspire the participants to explore the new opportunities in pharmaceutical research and the translational science. And with these words, I welcome all the participants to actively participate in this roundtable discussion on the drug designing, discovery, and development. And I encourage and I request everyone to actively participate, ask questions, and share your perspectives or the meaningful learning, because we know that this is the platform for the discussion and the exchange of ideas so that we can learn together. So thank you once again for joining us, and let us now proceed with the roundtable discussion on drug designing, discovery, and development. So now I request everyone, because we are only the 17 in number, to introduce one by one so that we can know each other. So let me introduce first our roundtable discussion committee expert, Dr. Prakash Salami sir is present here. So he is senior scientist in CFTRI Missouri. Our roundtable discussion committee member, Dr. Anand Dave sir is present here. So first of all, I would like to request you, sir, Dr. Anand Dave sir, to introduce yourself to all the participants and hello.
Yes,
Are
sir.
you
Yes,
there?
sir.
Yes.
Yes, yes, I'm here. So, good evening, one and all. Myself, Dr. Anand Dave. I'm head and assistant professor in Department of Microbiology and Biochemistry, KK Jarudala Maninagar Science College, Ahmedabad. Uh, since my working area is agriculture microbiology, PGPR, gut microbiota, probiotics, and all. So, welcome to all the PhD scholars and other faculty members who are here. Thank you.
Thank you, Anand. Then the Subhrajyoti Sarkar, Abhishek Bhairav, Matri Datto. Just no need to open your camera, just unmute yourself and just you have to introduce yourself, that from which institute you are. And you're a student or the PhD scholar or the faculty member. Nitin Jalake.
Good
Hello.
evening, sir.
Good evening, Maitri Datto.
I'm Maitri Lata Sargiari, and I'm working as an assistant professor at Assam Don Bosco University.
You are working on the same field, madam. Your research interest on the drug designing.
Yeah, no, mine is life sciences. My specialization is entomology,
Thank you,
but
madam,
I'm
for joining.
interested in drug designing.
Yes. Thank you, madam. Abhishek Vairo.
Good evening, sir. I'm Shilpa Savargi. I'm a research
Good evening,
scholar
Silva.
from KIST, KVV, Karad.
Thank you, Silpa, for joining. Ashwini Patil. Please unmute yourself, Ashwini Patil. Your camera is on, but your audio is mute. Ashwini Patil. Nitin Jalke.
अश्विनी बोल रही है अभी.
Good evening, sir.
Good evening, good evening. Good evening, Ashwini.
अश्विनी
I think the network issue. Yeah, now you are.
Hello, good evening, all.
Good evening.
My name is Abhishek Paron, an MSc Microbiology from Amity University, Lucknow.
Oh, thank you.
My interest in immunology. Also, I have joined the drug discovery seminar.
Shall we learn something in seminar? You can share your experience in seminar here. What you have learned from the seminar, you can share here, basically.
So right now, I have joined the first seminar of drug discovery. This is the first webinar I have joined
Oh.
for the discovery.
Thank you, Abhishek. Dr. Sahajada J.R. Rabani. Dr. Sahajada. Mariam Saleha.
I think Pooja Kambod is also there. Pooja Kambod.
Pooja.
Hi, hello, good evening.
Yeah. Good evening.
Yeah, it's Mariam Saleha.
Good evening, Mariam.
A very good evening to all of you. And I'm Mariam Saleh, and I'm pursuing MSc Microbiology from Jamia Millia Islamia, New Delhi.
So thank you, Mariam. Your audio is not audible. You are not audible.
Hello.
Hello.
Yeah, am I audible now?
Yeah, you're audible.
Yes, sir.
Oisi Biswas. Are you there, Oisi?
Yes, sir. Good evening.
Good evening. From which institute you are, Oisi? Just introduce you.
Sir, I am a second year student from
Okay. Thank you, Oisi.
Xavier's College, Park Street, and I am studying biotechnology.
Now, Parakram Singh Chauhan.
So, good evening, sir. My name is Dr.
Good evening.
Farhata. I'm working as
Yes.
a assistant professor in Chitkara College of Pharmacy, Chitkara University, and I'm also a state vice president of MBSI Punjab, and my
Okay, so
research
great, sir.
area is pharmaceutical microbiology, biotechnology. That's it.
Okay.
Protein engineering.
Thank you so much, sir, for joining. So,
Thank you, sir.
Prakram sir,
Thank
your specialization in pharmaceuticals, so you can share your experience with the participant.
Yeah, sir.
It is
Actually,
related to the drug design.
I completed my MS Pharmacoinformatics from Naipur Mohali. Then I
Yes,
did
sir.
some molecular modeling and docking study between enzymes and receptors. So, and I use Autodoc, Modeler, QSAR. So I have little bit experience of bioinformatics.
So I request you to kindly share your research experience, sir.
What, sir?
You can share your research experience here, sir. Means on
Okay.
what were the targets and which type of the compounds you have selected as a drug?
So basically, sir, we use the enzyme ligand interactions. So our protein is basically an enzyme, and for that we try to find out the which of the best substrate for them to enhance the activity. So, like for example, there is a one enzyme called xylanase and manganase. So for that, there are different type of oligosaccharides is used as a substrate, like monobios, dios, tetrose, pentose. So we screened all these things. And also we did molecular modeling between phytochemicals and their targets, like moringa. In moringa, what you can say, moringa is commonly called as drumstick. So we isolated a compound from the moringa leaves, and we did some antibacterial study. And we tried to find out where this aromatic compound actually bind to the bacterial site, whether it is a protein inhibition or cell wall inhibition. So we find out that it's bind on the 30th subunit of the gram-negative bacteria. So we all these things in bioinformatics.
So you have done, sir, your molecular docking by help of Autodoc only.
Autodoc, and also we did modelers.
Modeler, that for the modeling of the.
Yes. And for docking, sir, we did collaboration with Naipur Mohali. So they have state of art of all the bioinformatics software.
Thank you, sir, for answer.
Thank you, sir. Thank you.
Now the Rusi Misra, Pooja Kamboj.
Hello,
Yes, yes.
good evening to all. My name is
Good
Rushi
evening.
Mishra, and I'm a second year biotechnology student at Sumaya Vidya Vihar University, Mumbai. Thank you.
Okay, thank you, Rusi, for joining us. Pooja Kamboj.
Sir. Good evening, sir. I am
Good
Pooja
evening.
Kamboj. I have completed my master's in microbiology from Guru Jambeshwar University of Science and Technology. Yes, sir.
Thank you, Pooja.
Thank you, sir.
Nitin Jalke.
Good evening, sir. I am Nitin Jalke.
Good evening.
I am
Good
working
evening,
at Serum Institute. I am working at Serum Institute of India, Pune, in microbiology department.
Okay. So have you worked over this drug designing part?
No, actually, I am working
Okay. Your voice is cracking, Nitin.
Hello, sir.
Yes, Nitin. Now you're audible.
I am working in quality control microbiology department. Samples came from production department and we test it, like water testing, bacterial infection test, and sterility testing, like all these tests related to microbiology.
Okay.
Okay. Thank you, sir.
Thank you, Nitin, for joining. So now Upasna also joined. Are you there, Madam Upasna?
Yes, hello.
Yes, madam.
Good evening, everyone. I am Dr. Upasna Chetri, currently working as an assistant professor in the Department of Botany, the Assam Royal Global University. And I feel today's topic, drug designing and discovery, is very relevant in the face of AMR, that is antimicrobial resistance, where we are looking for new drug and new drug targets in microbes. So I hope this session will be a very fruitful discussion on this current topic.
Thank you,
Thank
madam.
you.
Shivani Kumari. So now we have introduced Dr. Prakash Salami, sir.
Yes.
Yes, sir. We'd like to now hear from you, sir.
Only you or other participant also?
Other participants, I think, also.
Yeah, actually, I started some other work parallelly, but then I think I can tell. With reference to the cell biosensor that we use for screening antibiotics, for detecting the, for identifying their mode of action, right? But any drug you develop, especially the antibiotics, not the other drugs, which we may be talking. Here I am talking only with reference to the antibiotics. So we know that they will either inhibit one of the pathway, either cell wall or the protein or DNA biosynthesis or fatty acid or RNA biosynthesis. And then we have actually the such pathway specific genetically engineered bacteria. So this bacteria, what they do is that they senses actually in a very nanomolar concentration of antibiotics available in the environment and induces the beta-galactosidase gene. So this actually facilitates the high throughput screening, more space identifying the antibiotics directly for their mode of action, and you can actually do in a shorter time. So through this method, beside identifying the cell wall acting antibiotics are like Nisin or Subtilin and their variants, we could identify bacitracin producer, and more importantly, the the RNA biosynthesis inhibitor, that is lasopeptide. Otherwise, this would have actually taken at least a years together. And this lasopeptides has a very similar activity like a rifampicin. So this is actually the present work is going on in my lab, where the lasopeptide from a bifidobacteria as well as a bacillus we are being characterizing. And bifidobacterium lasopeptide manuscript paper just got some 10 days back accepted in Current Microbiology, where we have shown through the genomics that it has a conserved gene gene operon, and has able to produce, can produce the lasopeptide, which we have actually functionally analyzed, especially with reference to the Tricin SDS page and antimicrobial activity, particularly against the Mycobacterium mycoid. No? Because Mycobacterium mycoids is a non-pathogenic mycobacteria. Now we want to take up this work further to develop a drug against drug resistance mycobacteria. And now there is a call for proposal from ICMR on exclusively on mycobacteria. Means how to treat the tuberculosis. I think this month end is the deadline to submit the project proposal on tuberculosis related drugs. So we are actually working on that. So this is with reference to from the Bifidobacteria, and then Bacillus, actually we have cloned those genes associated with the lysopeptide production in E. coli. And then E. coli actually is producing the lysopeptide. And then we are going to scale up. So in E. coli, we made a synthetic construct. And through PCR approach, we clone the gene. And then now those genes are now actually expressed by E. coli. So purification is becoming much more easier in a larger scale. And then in addition to that, once that intact clone is there, then it will be easier for us to determine the different site, the specificity of few of the amino acids, then roll up the conjugate sequences, the active site, and all these kind of things. I will supplement further, or depends on this interaction, then I can answer few other questions also. So our drug discovery are basically the antibiotics in this case are from the natural sources, especially from the fermentation broth or from the environment. If those cultures are producing the antibacterial substance, antimicrobial substance, how one can identify rapidly for their mode of action. So suppose you give me tomorrow some sample, either plant extract or a fermentation broth or some any extract, soil extract, and tell that it has a antimicrobial activity. It shows the zone of inhibition. Can you can ask me in how many days I can tell you what is the mode of actions. So I can tell you what is the mode of action within a 1 day. That is what I am trying to tell. Okay, without purification, without characterization, and with a minimum sample. So this is with reference to the antibiotics. The other area of work which is going on the postbiotic in our laboratory, where we are looking for again the fermentation broth or the cell wall or the postbiotics molecule from the some of these grass organisms which have alpha amylase inhibitory activity, alpha amylase and alpha glucosidase inhibitory activity. Now, why we need alpha amylase? So that there will not be a rapid hydrolysis of the starch. There will be a slow hydrolysis of starch, and where it is needed, especially for the diabetics patients, right? So based on those lead available, we are actually looking for a functional food where this postbiotic molecule will be incorporated in the form of a drug or as a functional food in this case, and then diabetics can be controlled. Okay, so these are the such a two areas are there at this moment. The areas I will talk maybe a little later. Thank you.
Okay. Thank you so much, sir. So now,
Sir, may I?
sure, sir.
Sir, lactic acid bacteria having all these characteristics, alpha amylase activity and all these told us.
Yes. Yes, you have to have actually a bigger screen, means more number of sample, and then a rigorous analysis. Then you will be able to differentiate, and then you should have the positive control. So once you have the positive control, your result should match or should be equal to the positive control, then it is possible. So we use a strong positive control, and then we compare with that one. Yes.
Okay, fine, sir.
Yes.
Thank you, sir. Thank you, Shilpa. So encourage everyone, if you have any not research specific, if you have any knowledge on the drug designing, you can share here. Please unmute yourself and you can share any knowledge or your research insights or your experience in this way. And if you have any question regarding this drug designing, may I ask? Abhishek, Ashwini, Dhanya. Because this one is the platform for the discussion. Also, Prasun sir has joined. Prasun sir, are you there, sir? Prasun sir.
Yes, sir.
आप ही का वेट कर रहे थे सर हम लोग.
Sorry, sir. Today I was a bit busy. Just got time to join. Yes, sir.
Just share
So,
some things on this topic, sir. Drug designing today's topic, sir.
yes. No, I think it is a very important topic, especially when we know that antibiotics, we don't have much antibiotics in the pipeline to tackle against MDR and XDR. So when there is very few drug in the pipeline, we need to think uh, some other way or use AI tools to screen as many molecules as possible or find new drug targets. For all those things, I think this approach is better, computational approach to begin with, followed by the testing. So conventionally, we know that to reach a drug to the market, it takes minimum 10 years if it is done carefully and successfully through wet lab. But nowadays, with the help of computational tools, we can at least do the preliminary screening a bit faster to get some lead molecules. And then there are several plant extracts and so many other compounds that are available and still remaining to be explored. So
Even say in
those
2003, yes, sir. Sorry, sir.
yes, sir. No problem, sir. Yes, continue.
Even in 2003, sir, one study was done. Okay? They have screened drug by both methods, by the wet lab, conventional one, and by the computer drug, CAD. Okay, but they have come up with the same drug, sir.
Hmm.
In the
So that's
conventional
the
method,
potential of competition tool, that it is
yeah,
quite accurate. Although definitely during trials we have to be very careful whenever we
sure.
take help from computer. Hmm.
So anyone, thank you so much, Prasun, sir.
How to go for first step?
Hello. Yes, Silpa.
What is the first step for that?
For what purpose?
Like sir said, I didn't get it.
Okay, the first step in case of drug discovery, the first step is first is what is your objective of the study. Okay, suppose you want an anti-malarial drug. So you want to inhibit the growth of malaria or just infection. You want to inhibit the infection at the preliminary step itself. So you identify the pathway through which the pathogen is infecting and how you want to stop that. So there must be some kind of a speculation that you are going to target a particular membrane protein or some internal protein or other biomolecule biosynthesis so that it don't cause virulence. Okay, so the target can be anything, and based on that, you are trying to develop a drug. So once you fixed the target, then you can screen some drugs. There are several approaches. So in case of in silico approach, there are uh, approaches like you can go for only FDA approved drug. So you can repurpose already existing drug that is already certified for FDA. So by doing so, you actually covers one additional step, that since the drug is already FDA approved, so getting clearance at the clinical stage will not create much issue. Okay, but all these things are theoretical. Then the first step, like you asked, is to identify what type of molecule can target the protein or the molecular pathway. Okay, for to do that, you can use Autodoc, and there are several online platforms as well as proprietary platform also, like Schrodinger's software you can use to dock two molecules, check the binding energy, the lowest binding energy, and other rules that has to be followed in order for the drug to be effective. Like there is Lipinski rule, Lipinski rule. Okay, so it has to follow all the criteria to be categorized as a drug. And then
Okay,
there
sir.
is some institute also have the facility. So you basically have to use software like Autodoc Vina, or Schrodinger software to dock two molecules, three molecules, or maybe multiple molecules against one protein or maybe 10 protein is your target, and there are 1,000 drugs that you want to check or screen. Okay, out of 1,000, how many drugs are having lowest binding energy and are fitting to the criteria? But you cannot do this on your simple computer. For higher kind of a study or a higher screening, you need supercomputer. So there are GROMACS. GROMACS is again a platform that will run on supercomputer. So some of the institute also have supercomputer facility where they can allow you to use it for 1 week or 2 week. So the whole supercomputer will run for maybe months if you are screening more compounds, or if you are too stringent, you are keeping the parameters for your screening too much stringent. Okay, so in that case, you need supercomputer so that you can screen such compounds.
So for plant extract, the same procedure is there.
Yes, same procedure. Again, plant extract are mixture of compounds. So among those, what are there in the mixture? So you may do the activity text directly through wet lab experiment. So you know that, okay, suppose methanolic extract is working very fine against your pathogen. So what are there in the methanolic extract? You can get to know through mass spectrometry. So you can go for MS, check which compounds are present there. Then most likely among these 10 or 20 or 30 different compounds that are found in mass spectrometry, any one of them or maybe a synergistic effect is happening. So it may be based on the concentration, okay, but you need to test them one by one. So either purify those compounds and test in wet lab directly, or use the help of computer. You means take the help of computer and screen again. Among these 30, which one is more effective and how it may be acting.
Okay, sir. Got it.
Just I would like to add one thing here, Silpa. Suppose you have gone through that GCMS. Already the person sir has mentioned, and suppose there are the 20 compounds, okay? So you can see that the relative percentage, okay? So there will be the 2 or 3 compounds, the higher relative percentage, means abundance that is, okay? So first you can go for the molecular docking with only the 2 or 3 phytocompounds and see that binding energy. So based on that, and then we can go for the separation of or isolation of the compound. Okay. Means drug designing means computed drug designing will save your time only. But eventually, finally, you have to go for the wet lab experiment.
Yes, sir. Thank you.
So
Any
I
more
just want,
question? Because
yes,
sure.
I just want to ask you. You told about this binding energy. So this binding energy, when we work with the phytocompounds, we sometimes do not get above 7. If can they be, if we get some around like -6.5 or 6.9, can we go for drug designing with these kind of compounds, or always we should be like higher, more than 7? I just want to ask that.
Means -7, madam.
Yeah, if we get some phytocompounds which are showing good, means they're showing docking properties. I do not know if it is good or not, if it is -6.5 or -6.9. Can we go with those compounds for drug designing or not?
Prasun sir, are you there, sir? I think that
Because
discussion
few members say that our binding energy should be more than -7. So I just want to ask you about that.
Yes, with lowest binding energy, it is always means there is a good chance that the drug will be effective. But there is no, so it is a kind of threshold that people have been repeatedly doing, and they got to know that, okay, uh, below this much of energy, so suppose -7 is the cutoff, people have been practicing. But even you can see some studies going for -4.5 also, they are finding that drug effective. Okay, so it is binding energy is a kind of calculation that the software gives to you based on the docking. Okay, the interaction between different atoms present within the vicinity or the active pocket you decide. Right. But ultimate efficacy will be checked only through wet lab experiment. So if you are observing
okay.
that in wet lab experiment the compound is working fine, then you
Yes.
can also choose lower binding energy to show that docking through docking you can show that okay it is binding here and this is how it is inhibiting. So if you are trying to answer that question of how this drug is inhibiting something, for that you can go ahead with lower binding energy also. But if you are screening, better keep universal threshold, okay, so that the work will be accepted across the scientific community.
Okay, and one more question I have, like when we go for this drug designing, like is it necessary to have an inhibitor, to search for an inhibitor? If it is, then how to go for searching these inhibitors?
Again, inhibitor studies, I'm not much aware, but it follows the similar pattern. But inhibitors may also bind allosterically also, no? So they can
Mm.
bind elsewhere on the protein, but
Mm.
they can do the job of inhibition. So they will alter the active site. For that, you need a good protein structure, and then the MD simulation will only tell the whether the inhibitor will work or not. So without
Okay.
MD simulation, it will not be possible to get to some conclusion. So after docking, there is a simulation that you do in a kind of physiological environment that is called molecular docking simulation, no MD simulation. So you need to go with that to conclude something.
Okay, then I just want to ask you, like if we get a minus 6.5 with binding energy with phytocompounds, can we go for MD simulation soon after the molecular docking, or shall we go for wet lab study after molecular docking
So
and
if
then
you
go
have not
for
gone for
MD
wet lab study,
simulations?
first go for wet lab study. Okay, if
Yes.
you are not sure on the molecule, then
Okay.
because further, and if you only have just say one molecule, then
Yes.
I would not suggest to go ahead, because it may be risky to go ahead. For MD simulation also take time, no?
Okay.
Time, and
Means
again
you are
you may
talking
be
molecule,
wasting
means you are talking about the phytocompounds, sir?
phytocompounds. Yes, yes.
Yes.
So if you
If
have
I
only
have
one
got one,
phytocompound, one target
huh?
compound, then it
Okay.
is like you are trying to hit one thing with one pebble, right? If you have 10 pebbles and you know, okay, at least one may hit the target, so that increases the chance. Okay, so
Okay.
just
So
through chance
so
wise,
how
it is
many?
not like it may not be
Hmm.
successful. It may be
Okay.
successful, but uh, in computation, as computational aspect, we always go for chance or probability that whether it is good or not. That's why this kind of
Oh,
threshold people have kept to avoid wasting time.
okay. Okay. So if it is within the threshold, and we have got minimum, how many compounds should we take for these docking simulations?
See, theoretically it is as many as possible, but
Hmm.
practically, so once you get the lead, see, we can't go in a dark path, no. So blindly we can't follow. Say
Yes.
I said one compound and it
Hmm.
must be effective. Okay, there
Hmm.
must be some logic. Okay, some
Hmm.
background work, some scientific uh, backbone should be there to support that. Okay, this molecule may
Hmm.
be affecting or maybe inhibiting that protein or something like that. Okay, so
Okay.
if there is enough evidence, either wet lab or maybe literature support, then you can go ahead. Okay, then even 10 is better. Okay,
Okay. Okay.
but
Okay, sir.
we can't exactly define how many. Okay, uh, if you go for ADMET analysis, you may
Yes.
have seen in several other studies, people go for ADMET analysis, they start with 10,000 molecule or the whole database. Okay, they
Yes.
screen for some molecular cutoff, and then
Yes.
based on some parameter or threshold values, they will finally
Yes.
select
Yes.
some 200, 300 compounds, further
Mm-hmm.
narrow it down to maybe just 10 or 15
Yes.
and
Yes.
17, like that. So
Mm-hmm.
anti-cancer compound, anti-malarial compound,
Mm-hmm.
people go like that. Okay.
Okay.
Yeah.
So now again, since you have talked about this ADMET, so there are many parameters there. So which are the parameters mostly do we consider for our studies? And one more question, another question is with EDMED, like should we, before going for the molecular docking studies, should we screen out through the EDMED, or after molecular docking we need to go for these ADME properties?
Okay, so I think if you are just screening without wet lab, you should go for admit first. Okay, based on the target that you have decided, no solubility, what kind of solvent it may act on, or what kind of environment this drug will go in future. Okay, if it is for gastric patients, so acidic pH it should be working fine without losing its structure. Okay, so all those things, what are the target? Based on that, first you go for admit and then you go for docking. Docking is interaction of two or more than two molecules at a time, right? So docking you will go once you screened or narrowed down your search. Okay, regarding the parameters, I will share some other time because I don't exactly remember all the parameters and I need to give some exact values. No,
Yes,
so
sir.
we may discuss it, or I will share with you in maybe next meeting if I join
Okay,
in similar
okay.
thing. That time
Okay.
we can discuss.
Okay, and another question, just actually I was working with something these days. Then I got a very good docking score with two compounds. Okay,
Mm-hmm.
so one compound, like it was like minus 8.9, and I did not say I'm not so good in all this, but still I feel that since it is more than minus 7, it is one is minus 8.9, another one is also around like minus 8.9 only. So I got these two compounds at the same binding energy. Then now the thing is that when we follow the Lipinski's rule, one of the compound, it is within that Lipinski's rule, means
Mm-hmm.
that lipophilicity and all these properties. Okay, and another one, the molecular weight is, I have found it has got the same binding energy, minus 8.9, minus 8.9 with the other one, but the molecular weight is more than 500. It is around like 540.
Mm-hmm.
So can we consider that or not?
Yes, minor
It has a
minor
good
gap you
binding
can
energy.
consider.
Hmm. The
Yes, yes.
same binding energy, but it
Only
is
the
more than
molecular
that.
weight is higher, right?
540 or 600 like that, not more than that.
Mm-hmm.
Yeah, so can we consider that or not? My question is
Yes,
that
yes,
because
yes. You can
within,
consider.
if it is below 500 or 500, according to the rule, it is within the Lipinski's rule. But my compound is like I got around like 600 or 590,
Yes,
something
yes.
like that.
See, you got two lead molecule. I would
Yes.
suggest you go for MD simulation further. You do the MD simulation. Okay, get
Yeah.
the uh, get the data, and
Yes.
based on that data, you can decide. Okay, minor change in the molecular weight should not means hamper much.
Okay.
Okay,
Okay.
so that is a minor change, and it is also like molecular weight of the drug is also
Yes.
related to if you want to pass the drug through blood-brain barrier or
Hmm.
the target. Okay, based on the target
Yes.
also you have to consider the molecular weight. Okay, so
Yes.
if the target is not something like that, or
Hmm.
if it is a just topical application, then you
Okay.
can go ahead with this molecule as well. Okay.
Okay. Can you give me some more ideas about these molecular docking simulations?
MD simulation. So there are tools already. I told you there are tools available. So computational
Yes.
tools are existing. You prepare your molecule.
Yes.
Like once you have decided that okay, these two drugs may interact through a autodoc, then you
Okay.
simulate in a physiological environment where pH is fixed, salt balance is there. Okay, like a
Yes.
physiological environment we consider. No, so say for
Okay.
example bodily fluids like blood or saliva. So you give that environment. So in MD simulation,
Mm.
all other molecule, in case of docking, there is no other molecule, only two molecule is there, your target molecule and the drug.
Yes.
But in MD simulation, there are presence of other water molecule, some salts, and pH for pH balance. Suppose if it is a blood environment, maybe things that may be present in the blood you want to keep. Okay, to
Okay.
simulate that environment, like it is a simulation, right? So to
Yes,
simulate that environment, that physiological environment, there will be other molecules, and now you allow the same two molecule that you have docked already.
Yes.
Now you allow in that type of environment, because now the drug may interact with, say, some salt molecule, and it is not going to bind to your protein.
Okay, sir.
So if MD simulation result fails, then the drug fails. Okay, because it
Okay.
is interacting with some other protein, but not your protein. If your
Okay.
drug is too much specific to your protein alone in the simulation environment, then only the drug passes the test and it goes to the next phase.
Okay, sir.
Yeah.
So, sir, for the publications and also, is it mandatory to go for MD simulations? Otherwise,
Yes,
till
yes.
docking,
Nowadays
it is
docking
not.
nobody prefers, na.
Yes,
So
yes.
not even MD simulation, you need to go to even milliseconds. Okay. Previously, people used to go for just nanoseconds study. Okay,
Okay,
but
sir.
now people are asking more and more data, more and more
Okay.
number of repeats, or the confidence on the data should be more. So more computation is needed like that.
Okay. So where can we go for these MD simulations? So, do you do any MD simulations, sir?
In my institute, other people do. I don't do directly. So I have some collaborators. They are working on supercomputer. There they have the facility to do the simulation on GROMAX platform.
Okay.
Okay. Yes.
So if I want to do like, can you connect me to those people who
Sure,
can
sure,
do MD simulations for me? Okay, sir.
sure.
So how will you share the contact with me, sir?
I can share my mail ID via chat box. I am
Yes.
typing
Yes.
already. Okay.
Yes, sir. Thank you,
Okay,
sir.
so you can email me and then I'll let you know.
Sure, sure. Okay, thank you, sir, so much.
And thank you, Prasun sir and Maitri madam. So, is there any query more? Hello.
Hello. Yeah, I would like to ask you one thing that if I am a microbiology student and I want to learn drug discovery and drug designing from the scratch. So first of all, what's the difference between two and how do I learn it and equip it? And then if I want to like do my master's dissertation in it, so what's the criteria of it and what skills do I need to learn?
Prasun sir.
Ji sir, I think you are the better person here because it is a general about microbiology. But I'll give the input. If I understood her correctly, she want to know how she can learn this. She is a microbiology background and she want to learn about docking, right? Docking and all other techniques. So
Yes,
there are
yes,
some
exactly.
courses. There are some courses Dr. Viprant sir already run in between. Okay, 5 days course is there to begin with. Okay, and before going for any type of course, you should decide about your target work. Okay, say as a student, now you are very much interested in maybe cancer or maybe diabetes or something. So define some good project that you want to carry in your life. Okay, work on that because whatever you will learn, anyways, it is going to be helpful in all the fields. Okay, let it be immunology, cancer biology, or cell biology, whatever. Okay, but if you take interest in your own topic, then you will learn faster. Okay, because you will remain curious throughout the class. Okay,
Yeah, yeah,
and
I
where can you learn?
understood.
So, like Schrodinger software, Autodoc software, I told you, Gromax, each and every of the company and this software, those who have made it, they have also kept a tutorial on their website. Okay, so you can learn from them also through the tutorials. There will be a sample data that you can run. Okay, they will also tell stepwise what you need to do. But if you cannot understand by yourself, then you can go for some training program that is being conducted online or offline. So Schrodinger in Bangalore office, they also conduct training program, but they ask some payment, I think 15 or 30,000 rupees for that training. But they will give you that training is offline. So you have to go there and get trained. Okay, some of the training modules are online, but others are offline.
Mariam, I have shared my mail ID and the WhatsApp number, okay?
Okay, sir. Yeah, it's in the chat box.
Yes, yes. I have shared there, okay? My WhatsApp number and the mail ID. So if you need any
Sure, sir.
help, you
Thank
may
you,
mail
both
or
of you.
so any more question or query or anything that you want to share? Thank you so much, sir, Prasun, sir. You have enriched today's session.
Thank you, sir. Thank you so much.
So now, Prakash Salami, sir, had promised to us that he will share his experience at the last. Prakash Salami, sir, are you there, sir?
Yeah, I already explained about the cell reporter assays, and then I was talking how postbiotic can be used for as a specific, potent molecule for against the hyperlipidemia and diabetics, and the screening those from the natural sources. And once you have such molecule, then you can do the bioinformatics and then see the target specificity one side. And then parallelly you purify that substance and then you characterize, So this is what actually the presently work which we are doing, And I already explained about the new novel antibiotics which we have identified using the cell reporter assay, which is more specific, sensitive, and time saving. Right. So this is what from my side at this moment. Right.
Thank you so much.
Exactly.
So shall we continue this topic, sir? It's next week.
Yeah, it can be because bioinformatics part, I think there is a lot to understand. So we can either continue
Yes, sir.
or see some other
Okay.
task. Oh, not an issue.
So I have shared one feedback form in chat box. Okay. So please take your 1 or 2 minutes and fill out this one. Just check the chat box. I have shared one feedback form there.
The same topic we're going to continue next week, sir. Drug designing and discovery, right?
Yes, so we will decide, sir.
Okay, fine.
Okay, thank you. So thank you everyone for joining today's session.
Thank you. Thank you, everyone.
Thank you, sir. Thank you.
Thank you,
sir.
thank you everyone. Thank you, Prasun sir, Anand sir, Dr. Prakash Salami sir, Mariam is there, Matri madam. Thank you everyone for joining today's session.
Prasun sir gave very good response, sir.
Yeah.
Drug design discovery, sir. Because though I am also, I also don't have a much idea about the drug designing. So I use in my MSc dissertation on cancer, Autodoc and PatchDoc server I use, online server.
PathLab is the open server, sir. No?
Open server, yeah. Okay, fine. So all of you.
Okay, thank you everyone. So the certificate should be issued if we have filled out that feedback form. So certificate will be issued to all the contributors, okay, to your mail ID. One Kumar Arbind.
Somebody join, I think,
Hello.
later on. Will CMO,
Thank you, sir.
I think, Xiaomi 24115RA ATG. Somebody join, I think, but name is not there. Xiaomi.
Kumar Arbind. Okay, thank you everyone.
Thank you, sir.
Thank you, Mariam. And you may contact Mariam, okay? Either we can mail or you can use my WhatsApp number, okay? I can help you. that how to
Sure,
start from
sir.
the scratch.
Yeah,
How
yeah,
to
sure,
start
sir.
from the
I'll
scratch, I
contact
can
you.
help you. Sure, sir.